All right, good morning, everybody. Okay, so today's lecture is really going to be a little bit of a departure from the kinds of lectures that we've had so far. Why? Because there is not a chapter in the book on modulating memory with exercise. I'm really going to be describing for you some contemporary experiments. Actually, we'll start out with some historical experiments. So this will be a kind of different kind of listening experience because you're not going to have the book to fall back on. But it really gives you, hopefully, my goal here is to give you a flavor for how a neuroscientist might build up a case for the questions to ask about this really interesting question of how do you modulate memory. And the more general question is how plastic is the brain? Again, going back to this general question of neuroplasticity, what can change the brain? And so I'm particularly interested in this topic because, as some of you know, this is part of the research that I do in my own lab. And I mentioned to you that I gave a TED Talk, a TEDx talk in Orlando on November 5th. That's up on the web right now, so if you want to go and look at it, you can look up TEDx Orlando and Wendy Suzuki, and you can see the talk itself. It's about 15 minutes long. And it goes over some of the work that I'll be describing for you today, but it's a much more directed talk focused on this course that I developed here at NYU called Can Exercise Change Your Brain, and how I developed that and some of the results that I found from that, which we'll be going over at the end of the talk. So you can use that as your supplementary homework as well. Okay, but before I get into the exercise and modulation part of the talk, I just wanted to go over one more time a very, very important concept that we focused on in the last lecture that I definitely want you all to understand. It will be on the exam for sure, and that is the concept of the Hebbian synapse. And this is a more complex subject. I just wanted to go over it one more time to make sure that anybody that had any questions, please ask them right now or at the end of class. This is one of my days I'm staying, so anybody who wants to ask me questions after class is welcome. This was a more complicated topic, but because we went from a theoretical idea that Donald Hebb came up with just from his observations of behavior and his theorizing about what might happen in the brain. So we went from this theoretical idea to an actual channel, to an actual receptor, the NMDA receptor that actually we found underlied this big theory that Hebb first came up with. So that's the first idea that I want to stress, that it started out with a general theory, no evidence for it, and we ended up with an explanation for this theory that came down to the features of this NMDA receptor. Okay, so that's the overarching idea that I want you to understand. Let me just go through the steps one more time slowly to make sure that you understand all the links that I want you to get. So here is the theory. The theory is, again, Hebb's law, developed by Donald Hebb, theorizing that synapses grow stronger. That is, you get higher output, higher EPSPs. That's what I mean by grow stronger. When the presynaptic neuron repeatedly causes the postsynaptic cell to fire. In other words, you need concurrent activation of both the presynaptic and the postsynaptic cell. Action potentials of the presynaptic cell, but also activation, lots of depolarization in the postsynaptic cell. And the maxim that came out of this is cells that fire together, wire together. We went from this theory to an actual example of what this might look like on a neuron, where you have information from the left eye coming in to a visual cortical cell. And in this case, the right eye has been closed. So you get lots of activity, concurrent activation in the left eye. You get lots of presynaptic activity. The large amount of presynaptic activity causes large amounts of postsynaptic activity in this cell. So that is following our Hebb's law, presynaptic and postsynaptic activity together. And the result of that is, the idea is that you get strengthening. Here's how the strengthening is demonstrated here. That is a growing of these synapses. But as we see, that this increase in activity could be happening in lots of different ways. So the question is, what is causing this increase in activity, this potentiation, just because you get presynaptic and postsynaptic activation at the same time? And here, the next step was, again, this is still theoretical, was there a possible example that we can see in the brain? And our example, does anybody remember what that example that we found in the brain was called? Our actual electrophysiological example of this Hebbian synapse. Anybody remember? Yes. Not NMDA, it was one step before. It was the kind of plasticity that we talked about. NMDA is a type of receptor. Okay, so what is this called? Long-term potentiation. So this was the actual example that they found in the 1970s that seemed to be consistent with this theoretical idea written here and shown here. Why? Because here they were giving individual stimulations of either the perforant path or the Schaefer collaterals. You should know those pathways from the pathways that we went over. And then what they do is they give a big old high-frequency stimulation called a tetanus. Lots of activity, lots of stimulations in a short period of time. That actually artificially causes both presynaptic and postsynaptic activity. That's what we said is needed to cause change. And oh my gosh, what happens? You get a change. That same individual stimulation that gave you an EPSP slope that was down here, now the slope gets much higher. That means a much sharper increase in the excitatory postsynaptic potential. And look how long this lasts. This is called long-term potentiation. Okay, so this part of the puzzle is the example. It's an artificial example, but it happens in the brain. Now, does everybody get that? No questions about that? Okay, now the very last step is asking, fine, we got this example. If we give this artificial stimulation, we can get a potentiation that lasts for a long time. Why? Why is this particular pathway, again, this is either the Schaefer collateral or the mossy fiber pathway, why is this pathway so sensitive to this high-frequency tetanic stimulation? And the answer is what you said. What did you say? NMDA. So the answer is the NMDA receptor. The NMDA receptor is a glutamatergic receptor. It works on the glutamate pathway. You know that glutamate is the most common excitatory transmitter in the entire brain. All your excitatory stuff happening in your brain, most of it is because of glutamate as a neurotransmitter. And glutamate has two key receptors. Okay, remember we talked about D1 and D2 receptors for dopamine. Glutamate has two receptors, the AMPA receptor and this key NMDA receptor. Why does the NMDA receptor give us this? And that's why we go here. So here are these two different kinds of receptors. AMPA receptors act on normal synaptic transmission when you don't get this tetanic stimulation. But here is this special NMDA receptor. And NMDA, you don't have to know this, it's just so you know, it stands for N-methyl D-aspartate, and it is a special, the particular agonist for this particular kind of glutamate receptor. You don't have to know this, just know it's the NMDA receptor. And this NMDA receptor, works in a special way. It doesn't work for any normal synaptic transmission. You get some neurotransmitter out in the synaptic cleft and you get activation. No, this is typically blocked with Magnesium. Magnesium causes a blockade of this receptor under normal synaptic transmission situations. But remember, we said that something special happens when you have both presynaptic and postsynaptic activation. Here's the trick. When the postsynaptic membrane is activated, is depolarized, what happens is the postsynaptic depolarization that happens with lots of glutamate activation here and lots of glutamate activation in this is a dendritic spine, in parts of the dendrites that are surrounding this. So this whole general area is being activated. That will increase the postsynaptic potential here. And that is the key to alleviate that magnesium block. When the postsynaptic membrane is depolarized enough, the magnesium just pops out. And now the NMDA receptor becomes active. So now we have the special receptor that is only activated when you have lots of presynaptic activation, spitting out neurotransmitter, and you have a high enough activation so your postsynaptic membrane is depolarized. Okay, so now we have the activation because the magnesium has popped out. Big deal. What does that do? Well, once the magnesium pops out, the NMDA receptor is sensitive to calcium. Calcium flows in, and the calcium will activate special second messenger systems. Particularly, we talked about the CAM-K2 second messenger molecular pathway in the postsynaptic membrane. Big deal, what does that do? The activation by calcium of the CAM-K2 pathway will then go and allow more AMPA receptors to be inserted into the postsynaptic membrane. So now, when I go back and give my normal synaptic transmission, I get even higher depolarization in the postsynaptic side. Why? Because I have more of the regular kind of AMPA receptors. Yes? Yes, artificially. So this is how it was first discovered, by this artificial tetanic stimulation. And all of this was worked out by giving this artificial thing to see how it happens.
So under what circumstances would this happen?
Great question. So she said, okay, you have this artificial thing, but how do we know it happens normally? When does this happen normally? And that exactly was the key question that many people were asking between the 70s and about the 90s. When first they were working out these pathways with the artificial stimulation and working out, these pathways that could participate in a normal potentiation, but how does it happen normally? And that was a very difficult question to answer because the hippocampus is so complicated. It's hard to separate out synapses, and it turns out that the best example and the first example of real live potentiation that happens normally was seen not in the hippocampus, but in the amygdala. What kind of plasticity did we talk about that happens in the amygdala? Fear conditioning. So fear conditioning, they were able to show that before fear conditioning, the stimulation of just giving a tone to the ear would give a smaller response in the amygdala. But after fear conditioning happened, that same stimulation would give a significantly larger response, and that would last for a long time. And it was sensitive to NMDA receptor antagonists. So yes, it does happen, and it took it was actually right here, Joe Ledoux's lab, who I told you studies the amygdala a lot, his group demonstrated that for the first time. It was a major, major discovery of a real in vivo example of this kind of plasticity, and it really validated literally 20 years of work on this artificial kind of plasticity. So that's a great question. That was a very important finding. Okay, any other questions on NMDA and the relationship between NMDA and this idea of Hebb's law? I'm going to be asking you what that relationship is. You should be able to describe it. You should be able to describe it in a paragraph. And you should be able to tell me what's so special about the NMDA receptor and possibly, and also what's different about the NMDA receptor and the AMPA receptor found in the same exact synapse. Okay? If you have any questions about that, please come and ask me at the end of class today. Unfortunately, one of the things that I needed to, that I had to cut out because we just don't have time enough for everything is the lecture on sleep. And there are really fascinating studies on sleep, but also fascinating studies on the relationship between sleep and memory. I don't know how many of you feel that if you study something and then sleep overnight on it, you have a better understanding the next morning. There's a reason for that. There is interesting and very important processing going on in the hippocampus that happens overnight during REM sleep. And so what I was hoping to do here is capitalize on the sleep that you've done between last Wednesday and today so that the information that I gave you last week is kind of sinking in. And this one last kind of going over it will really allow you to understand all of the levels of analysis that we went over here. So, but if you still have questions, feel free to come up at the end of class. Okay, so we got through this. And so now we're coming back to, again, our understanding from this very important patient, HM, and understanding from him of the clear importance of, in the normal patients, normal MRI scans, the hippocampus, the entorhinal, the perirhinal, and the parahippocampal cortices, these areas critical for declarative memory that patient HM was so impaired at. So a lot of effort has gone into understanding memory impairment and the contribution of these structures to memory impairment. But what I want to focus on today is possible ways that these structures and the functions that these structures do can be enhanced. And this general question goes back to, again, a general question of plasticity. What can make our brain more plastic in even a stronger way? And the way that early neuropsychologists asked this question was to ask, can the brain change in response to the environment? What does environment do to our brains? And these were psychologists studying at UC Berkeley. In particular, Rosencrantz, Bennett, and Marion Diamond, who I'll talk about in a second, looked at, this was one of the earliest studies in 1960, looked at the effects of what they called environmental complexity or environmental enrichment and training and actual memory training on brain chemistry. So what they did is they raised these rats. You can see they're different species or strain of rats than we typically use in the lab right now. It's the white albino rats that we typically use. This is way back in the 60s, but typical lab rats. And they wanted to know whether there was any clear difference in their brain chemistry or brain anatomy when they're raised in this, which is called an environmental complexity, also environmental enrichment, with lots of toys to play with, lots of other rats to play around with, versus these same rats raised in impoverished conditions with maybe one other rat in the environment, no toys to play with. And what they found were striking differences in the effects of environmental complexity on brain chemistry and brain function. This is Rosenzweig, and this is Marion Diamond. Marion Diamond was the anatomist that started to look at the actual structure of the brain in the environmentally enriched rats, and it was in Rosenzweig's lab that all of this took place. So they asked, what happens if you enrich the environment? Here's an impoverished environment. Here's an enriched environment. And I always like to show this picture people are still studying the major effects of environmental enrichment. It's kind of the neuroscience version of the Mozart baby. So what happens if you bring lots of stimulation, positive stimulation to the brain? And some very proud graduate student or postdoc posted this Disney World of enriched environments, where they were very proud of this multi-level structure that served as their rat's enriched environment. So you can do it in lots of different ways. This is fine, and you can see plenty of brain changes. But what brain changes do you see? Oh, I'm sorry. This part of this does come from your book, because Rosenzweig was one of the authors. He actually passed away recently, and so you can find this figure in chapter 18, just illustrating the environmental enrichment. This, you can see, looks very similar to his original picture from the 1960s, how they at UC Berkeley were doing the enrichment. But one of the most striking findings from this environmental enrichment experiments, and that really struck the imagination of many neuroscientists to come, was really discovered by Marion Diamond, who was a neuroanatomist looking at brain structure. She asked a very simple question. So here is a coronal section through a rat brain. Okay? I don't know if you've never seen a rat brain. A rat does not have the same gyri and sulci as the sheep brain. It's very flat. Here is the structure of the hippocampus. Look how huge it is in the rodent. And down here is the start of the amygdala. And up here is, these black lines are not real. I've drawn these black lines. This measures just the thickness of the outer cortex. How thick is it between layer 1 up here with no cells in it and the end of layer 6 down here? What Marion Diamond simply did is asked, how thick is the cortex in rats raised in these enriched environments compared to the impoverished environment condition? What she found was, if you raise rats in enriched environments, certain part of their cortices get significantly thicker. We're not talking about kind of mutant rats with humongous heads walking around. We're talking about small but significant and reliable changes in the thickness of the cortex. And not all areas changed. Visual cortex changed, presumably because there was a lot more visual information that the animals got, particularly compared to the impoverished condition. Somatosensory cortex changed. Why? Because they have a lot more interaction with lots of toys, ladders, and running wheels. And also motor cortex changed. Why? Because they were running around a lot more. So in certain areas, you got significant changes in cortical thickness with enriched environments. Increases in cortical thickness. What was that due to? Did they just have suddenly twice as many cells in these areas? No, it was not change in the number of cells. It was the increase in the number of synapses. That means your axons and dendrites in these areas are growing and shifting. And because you have to shove more of those processes in a particular brain area, you get a thicker cortex. And also significant increases in dendritic branching. So what part of the dendrite? Here is a drawing of a typical pyramidal cell in cortex that Marion Diamond was examining. And you have two different kinds of dendrites. Apical dendrites that come up off of the cell body, off of a main dendritic arbor. And then you have basal dendrites that kind of come off of the cell body itself. And you can ask, well, which dendrites are branching more? The apical dendrites or the basal dendrites? And here are the apical dendrites. Again, this is one figure from your text, so part of this is covered in the text, but it's quite small. I'm going to cover it in much more detail. We're looking at the mean number of branches per neuron for the order of branch. So the order of branch is, this is first order, and then when it comes off the first order, it's the second order. This is third order. So that's the order. And you can see that there's no difference in the number of branches as a function of dendrite order in either the enriched condition, the standard condition or the impoverished condition. No difference here. So apical dendrites, these dendrites up here, no difference. But let's take a look at the basal dendrites. Here, you see in the enriched condition, particularly in the order of branches, for the higher order branches, you get significantly more mean branches per neuron for particularly the third, is that the third or the fourth? 1, 2, 3 order branch, compared to the standard condition in blue or the impoverished condition in red. So specific parts of the dendrites are actually growing as a function of enriched environments. Now, not only, so enriched environments are a little bit vague. So lots of things are happening. We're not testing anything. We're not asking the animals to learn something. We're just saying, here, you have more toys, you have more stimulation, you have more social interaction. Let's see what that does to your brain. And the answer is, does lots of good things. Lots of growth, lots of more, many more dendrites. Another researcher, Bill Greenough, asked, well, I want to look more at learning, but kind of associated with enrichment. So I like to call these the Cirque du Soleil experiments, because what they did is they asked the rats to learn these complex kind of manipulations. So this is actually pretty hard to do, and he had to navigate over this and hop over the edge, because at the end of this he gets a treat, and he has to go over the ropes. And at first, these lab rats are really bad at it. They had to have little nets underneath it to catch them, and you know, just to make sure that they were safe. But after some experience, they got very good at this. I also have to say that you have to remember that we're talking about lab rats. These are not our NYU, not NYU, but New York City subway rats, which would, you know, run circles around these rats. They would beat the rats up, and they would jump over this without any problem, because, as you know, the rats can jump up on the benches, no problem. But still, for the level that these kind of more sheltered lab rats are at, this was the Cirque du Soleil kind of experiments made up a very strong kind of learning that they were able to do. And they got faster and faster at doing this. And what they found was also very striking brain changes. Now, this is both enrichment, but it's specific motor learning. And what they found was the motor learning provided extra brain changes even over rats that just ran the track without any obstacles to go over. So what did they find with these Cirque du Soleil types of experiments? So not only did they see increases in cortical thickness and synapse numbers, specifically for the motor areas now. They were focused on primary motor cortex, but they also found increases in numbers of blood vessels. Now, I should say that the increases in number of blood vessels was also seen in the environmental enrichment condition. That's very interesting. So can anybody think about why it might be good to have more blood vessels in your brain in general? Yes? Right. More nutrients can be transported in. Anything else? Yes? Oxygen, more nutrients and oxygen, both excellent answers. Any other reason? Think about in aging, what is a common brain deficit or brain kind of condition that happens to people that are elderly? So stroke happens, right? And stroke is a bursting of a blood vessel. And so the idea is that when you're young, more blood vessels can bring more oxygen, bring more nutrients in. But the more innervation you have with more blood vessels, if you do have a stroke in one branch, if you have more blood vessels, you have innervation to take over that. And that can help with long-term recovery. So the more blood vessels you have, the more innervation in general, the better off you are as you get older and get more susceptible to things like stroke. So these were not aging studies, but that's just an implication of more blood vessels. Increase in dendritic branching, we talked about that. Now, here is what was demonstrated with some of these motor experiments and Cirque du Soleil experiments. Increases in a molecule called BDNF. It's a growth factor, brain-derived neurotrophic factor, BDNF. What is this structure? What is brain-derived neurotrophic factor? Well, it was first discovered in development. So in development, you have lots of new cells being born. They have to go to the right places. There's lots of information, lots of growth happening in new neurons. And BDNF was first discovered in development as a molecule, as a factor that helps young neurons grow. If you remove the BDNF from development, you don't get the kind of growth that you typically need as brains are growing early in embryonic development. But, so let me just show you an example of that. So here is an example of the development of retinal ganglion cells in controls. So here are normal controls that are growing over a 24-hour period during development, and they start out stubby, tiny little axons, and then they grow and grow and grow, and they get very, very complex. Now, if in this situation you bathe the solution with anti-BDNF, that is a BDNF receptor antagonist, what happens is your stubby little neurons at 0 hour remain stubby and looks when this died at 24 hours. But if you give more BDNF, you get exuberant. This isn't necessarily good, but it just shows the power within development of BDNF to enhance plasticity, enhance growth of, in this case, the retinal ganglion cells. Okay, so that's what it does in development. But what about in adults? More recently, it's been found that BDNF also is a factor that is neuroprotective from ischemic damage. Ischemic damage is lack of oxygen from the brain. And it also potentiates synaptic transmission. So if you sprinkle some BDNF on a synapse, you get a higher activation of that synapse. In general, it's thought to be very helpful for plasticity and good to have around in general. Isn't it interesting? In environmental enrichment, you get increases in BDNF. Possibly, why? Because you're getting an increase in number of synapses and because you're getting increases in dendritic branching. And maybe BDNF is also there to help those branches find their right homes, just like it was doing within development. Okay, so BDNF is not just there, but people have done extensive studies of the effects of BDNF after a whole year of enrichment. So for a whole year, one group of rats was raised in enriched environments, and the other group of rats was raised in impoverished environments. And they looked at the BDNF levels in these two animals after this long-term raising in enrichment or impoverishment. And you see humongous increases in BDNF in the enriched environment group, again after a year, compared to the isolated environment group. So this is a factor that is changing enormously. This, I won't mention it, it's just so you know, it's NGF is nerve growth factor, a different kind of factor that's also increasing. Yes?
Does the BDNF drop off again if the rats
That's a good question. I am not familiar with any study that looked at it afterwards. Mainly, they've just demonstrated multiple times and really confirmed that BDNF is always increasing when you keep the rats in the enriched environment. But this suggests that more change continues to happen even at the end of a year. You might think, oh, you know, they get into an enriched environment and then they get used to it. It's like that becomes their normal. But this increased BDNF is maintained for a long time. And so it may suggest that when they go back, it continues to be plastic and it'll go back down. Unclear whether it'll go all the way back down to this isolated level, but that would be an interesting question. Yes?
How do they know that all this change is due to the enriched environment and not the combination of the enriched environment and the social interaction?
Yeah. So that's a great question. How do they know that this is due to the enriched environment and not more complicated interactions between enriched environment and social interactions and all the other things that might be happening in the environment? And that's, we're going to get to that question in a second. I'll tell you one very funny control experiment that they did. So they compared enriched environment to impoverished environment to what they called the TV condition. So rats in the TV condition could watch rats in the enriched environment, but they couldn't play in the enriched environment. And those rats were not enriched by that visual experience. But I always thought that was, I don't want to be in the TV condition, so I just wish you're not in the TV condition. Okay, so we're at the increased BDNF level. The other major finding in this study of enriched environments is the idea of neurogenesis. Now, we haven't talked about this before. It turns out that the hippocampus is one of only 2 brain structures where new neurons are born into the structure, even into adulthood. So you and I have new hippocampal cells that are being born, that get integrated into the hippocampal circuit. In fact, it's very specific. It's not all over the place in the hippocampus, only cells in the dentate gyrus. So you should know where the dentate gyrus is and what those axons to area CA3 are. So neurogenesis in the hippocampus only happens in the dentate gyrus. It's dentate cells that get born new in the hippocampus. The other place that new neurons are born in adulthood is the olfactory bulb. So it's unclear why that helps us in humans, because we really don't use our olfactory sense for that much compared to other species. But it turns out in both humans and in many other species, mammalian species, these are the 2 major brain structures where we know that adult neurogenesis takes place. Why is that such a big idea? Well, I would have to take you through the history of this. And for many, many years, starting in the 60s or 70s, there was absolutely no evidence for new neurogenesis in the adult brain. I don't know if your parents ever told you, don't drink and smoke because your brain cells, once they die, they'll never come back. Well, that's not exactly the case. They will, but only in the hippocampus and the olfactory bulb. For a long, long time, it was thought that no new neurons are born. After development, you have all your neurons, and then those are the neurons that you have for life. Has anybody heard that expression before or heard that thought expressed before? Yeah, so it's fairly common. But I'm here to tell you that there is neurogenesis, and it happens in the hippocampus and the olfactory bulb. So what does it have to do with enriched environments? Well, one very interesting observation was that raising rats in enriched environments actually enhances neurogenesis. So you get actually, you can get more new neurons born, and they survive for longer within the hippocampus. So this is just the definition, the birth of new neurons in the brain. And here is just a slide to show you that we can go in and look at individual brain cells that were just recently born. These are cells that just were born and inserted in, this is the dentate granule cell layer, the dentate gyrus cell layer. And if they get labeled with this substance called BRDU, you don't need to know what that is, but it's a substance that labels newly born neurons. You can look at how many neurons are born at any given time, and you can see this happening all through the lifespan of a rodent. And it turns out that BRDU is used to identify tumor cells that are multiplying. And so people with tumors get BRDU injections so the doctors can visualize a tumor, usually in your body, not necessarily in your brain. And so what a clever set of neuroscientists did is followed a set of patients that had tumors that received BRDU. And when those patients passed away, they were able to get their brain and ask, are there any cells labeled in the hippocampus indicating that even in humans you have new brain cells born? And this study that was done, I believe in the 90s, demonstrated definitively for the first time that this new neurogenesis in adults not only happens in rodents, but also happens in humans. So we're sure that this happens in humans from direct studies from these patients with tumors that happen to get BRDU that labels tumor cell division, but also new cells born in the hippocampus. So you get new cells in the humans, in the hippocampus, and the olfactory bulb. So this happens in mice, in rats, in monkeys, and in humans. But really, the environmental enrichment experiments have mainly been done, and the effects of environmental enrichment on BRDU have mainly been done in mice and rats, but we'll talk about a study at the end of the session where there's some evidence that, that things can influence the new birth of cells, even in humans as well. Okay, so we get increase in neurogenesis, and so getting back to your question, what is it about the enriched environment that is causing neurogenesis? Is it social interaction? Is it running around? Is it more exercise? And it turns out that a major function underlying a lot of the plastic changes that are seen is actually the physical exercise that the rats are doing compared to the impoverished environments. This is a major key. I'm not saying that the social interaction is not important at all, but you can get a lot of all of the changes that I just went over simply by giving rats, individual rats, access to a running wheel. Okay, so exercise alone can increase cortical thickness, mainly in motor areas, increase number in synapses in those areas, number of blood vessels in those areas, number of dendritic branching, increases in BDNF in the brain. Exercise alone can increase neurogenesis. And so von Praag and her colleagues wanted to try and compare and contrast a number of different conditions to figure out what was most beneficial for enhancing neurogenesis. So the first condition was the thing that we know that they knew enhanced neurogenesis, their typical enriched environment condition. Second condition is not enriched environments, but just the running wheel alone. I don't know if you can see, but you can look at it. This is a rat, 3 rats living in a cage, but with a running wheel. So they can hop on and off. They share the running wheel. And here is the one of the third conditions, which is rats with no running wheel living. This would be the isolated condition, enriched condition, and exercise condition. They had a fourth condition, which is a learning condition. This is a spatial learning condition. And this big tub of water, a little rat spa here, is called the Morris Water Maze. And this was developed by a man named Richard Morris to study hippocampal function in rodents. It turns out in rodents are very spatial animals. They have a very strong sense of their spatial environment, and that kind of spatial processing and spatial memory is highly dependent on the hippocampus. And Richard Morris developed a task that is very, very sensitive to hippocampal function, hippocampal spatial memory function in rodents. And you need to know how this task is run. Here is a typical pool in which this task is run. And you can see, can you see this darker dot here? This is a platform that is just on the surface, and it's labeled black, so rats can actually see that. But in the real condition, this platform can be placed anywhere, and it's white. There's powdered milk or non-toxic paint, white paint, that's put in the water so that a white platform just underneath the water is invisible. So rats get put into the environment. They can see the entire room, and they swim around until they find the platform, just swimming around by chance. And it turns out that normal rats are really good at learning where that platform is in navigating, using the cues of the external environment. Rats are very good at this, and this task and that particular version of the task is highly sensitive to hippocampal damage. So if you damage the hippocampus, the rats are terrible at memory for the spatial location. So here, for example, is another picture of a Morris water maze. The rat is triumphantly standing on the platform. He does not like to be in the water. And to motivate the animal, you can make the water a little bit cold, so they especially want to get out of the water. This isn't freezing water, but just gives them an extra motivation, and they will always climb up onto the platform. Now, in the behavioral and integrative neuroscience undergraduate class that I teach, that I have taught in the past for neuroscience majors, one of their labs was the water maze task. So we brought 8 kiddie pools into the lab. We filled them all up with water, put paint in them, and put white platforms underneath. And then when we put the rats in, and they kept swimming and swimming and swimming. Even if they touched the platform, they didn't swim. Turns out the water was too warm, and they loved it. They loved swimming around. And so that was a good lesson. We made it cold next time, and they did not swim around. And then they went and found the platform. So it really depends on the motivation here. But in cold water, they don't like it. It wasn't warm water. We thought it was like tap water, but they really enjoyed that. Anyway, this is a schematic view from top looking down. The square represents the location within the water maze. This is north, south, east, and west, where the platform, the hidden platform is put. And here is a very experienced, this is the path that a very experienced normal rat will take. If he's put in from this location here, he'll look around and he'll go right to the platform. That is a very short path length, and this is a mark of a very experienced normal rat to find a hidden platform. Here is the pathway that a rat with a hippocampal lesion will take after the same amount of experience as this rat. He will still keep going around. It's not like he suddenly likes the water because of his hippocampal lesion. He certainly wants to get out when he finds it, but he does not have the same kind of specific spatial memory that the normal rat has, and he's wandering around. He just does not know where that platform is. And so that was a little aside to tell you about a Morse water maze task. We're going back to this experiment where there are 4 different conditions that we're looking at to examine the effects on neurogenesis. What does enriched environments do to neurogenesis? What does exercise alone do to neurogenesis? What does impoverished condition, no exercise, just a couple of other rats around, do to neurogenesis? And what does water maze learning do to neurogenesis? Let's look at the results from this experiment. Okay, here we are. And now I have to tell you just a little bit more about how these neurogenesis experiments are done. I told you about this, this drug, BRDU. This is the drug that labels cells that are dividing. This is a drug that's used to identify neurogenesis. And so what you have to do is you can inject BRDU. It will label cells right at the time of injection, all the cells around this time that have just been born. And then you sacrifice the rat, and you can look at the number of labeled cells. And in this experiment, they looked at the labeled cells at 2 different time points. So they gave BRDU over a period, multiple injections over a period of several weeks, and then they sacrificed the rats 1 day after the last BRDU injection. So what we're looking at here is the number of BRDU positive cells. Remember, these are the number of cells that are newly born, that have just been born, new neurogenesis. These are all adult rats. But this measures proliferation, the early kind of birth of neurons in these different conditions. We have control, we have learner, that is the water maze animals. We have swimmer. Oh, sorry, I forgot to tell you that the other control was learner is just swimmer. So he just got to swim around for exactly the same amount of time that the rats that were learning swam around, but he couldn't learn anything. He just swam. He's a swimmer. Here's a runner with the running wheel, and then here's the enriched group. And so for proliferation, for the cells that are just being born at that time, look at the only condition that is causing an increase in the number of BRDU labeled cells. It is the running condition. The running is actually enhancing the number of cells being born during this condition. None of the others, there's no significant difference. So running causes an increase in cell proliferation. This is voluntary running. Oh, that's the other important aspect. So by running, this is voluntary wheel running. If you force the rats to run and don't let them get off the running wheel, that causes stress. Stress is very bad for your hippocampus, and you will not see any of these positive effects. So all of this is voluntary running. They looked at the BRDU labeling in a second condition. The second condition was 4 weeks after the last BRDU injection. Okay? So again, while the BRDU is being injected, all the cells that are born during this week time are being labeled, and so look at it right immediately. That's proliferation. 4 weeks later, those cells are still labeled, but some of them have died because some of the new cells that are born die. And so when you look at the number of BRDU labeled cells 4 weeks after the BRDU labels, you have a measure of new cell survival. Okay? So how many cells are surviving? So here, the result was very interesting. We also got an increase of survival with the runner group, but the same level of survival was seen in the enriched group. So running is doing, essentially, for the long term, is doing equivalent things to enrichment. So you don't need all the social interaction. All you need is a running wheel and voluntary access to this running wheel. And you get both increased proliferation, which might be doing something. But the main point of this study is, after 4 weeks, which group had the best kind of enhancement of survival of these newly born cells. And that was both the runner group and the enriched group. Okay? So this is one of the experiments that showed that a major factor in the enriched environment is this condition, having access to the running wheel, which is right there. Yes? Yes?
Why would the control group have a higher rate of proliferation than runners and swimmers?
They do not. Oh, between learners and swimmers? Yeah, so this is not significantly different. Yeah, only this one with the star is significantly different from the control, and these two are both significantly different from these three, basically. So yeah, I see that. That is a numerical difference, but it's not significant. Okay? Any other questions? So this is, I told you the idea, or I presented the idea that exercise is one of the key elements that is causing the brain changes, and this is one of the experiments that is given in support of that idea. Running alone can cause the same level of cell survival of newly born cells than the enriched, compared to the enriched environment. Okay? Okay, but what else can exercise do for your brain? Okay. So what we want to do now is kind of a combination of conditions examined in these experiments. Now we want to ask, okay, we know exercise is good. Can exercise actually make you smarter on a hippocampus? And exercise is increasing neurogenesis and proliferation. Exercise gives you more dentate gyro cells. Is that good? Is that good for your memory? And we want to test it on this hippocampal dependent task. So we're giving BRDU, we're giving running access or normal cage access. This is impoverished. This is the exercise condition. And then after access to either running wheel or normal cage, we're measuring their performance on the water maze. Again, remember I told you the water maze is dependent on normal hippocampal function. What we find is we're looking at that path length. So the lower path length is better. And we're also looking at latency. Shorter latency to find the hidden platform is better. So the lower the line, the better. Who's doing better? The runner or the control? Here we say both. We see in both path length and in latency, the runners are doing significantly better than the controls on this task. So this is one of a number of tasks, studies that have shown that running alone can enhance certain hippocampal functions, can make, in this case, the rat's spatial memory better. Okay, so we have this list of all these things that it does. Neurogenesis, it increases neurogenesis in the hippocampus, exercise alone does, and it improves spatial memory function. So that's great. These are some of the animal model questions that have been asked on for this question. But the big question that you might be asking yourselves as you're trying to learn, as you're trying to remember all that ton of information you're going to have to remember for your finals coming up in 2.5 weeks. What about me? What can exercise do for my brain? And so first thing I want to emphasize that in this study, I said in the beginning, we're not talking about brain growth into a mutant rat. And so somebody who exercises a lot, like Lance Armstrong, we're not talking about him turning into like a conehead person. But again, we could ask the same question. Can increased aerobic exercise, and this is a slightly extreme case, a very extreme case, can it actually change the structure of the brain so we can measure it? Now, we can't measure it in the same way that they do in rats with histology, but we do have structural MRI imaging. And the answer is that yes, exercise can do multiple different things that we showed in rodents, that we looked at rodents, and the answer is all good news. So, sorry, before I tell you about the structural studies, I want to tell you about the studies that were done mainly in the elderly population. I have to say that most of the studies done on the effects of exercise on brain structure and function have focused on the elderly. And this is because there is a well-known cognitive decline that happens in the elderly. And this cognitive decline is a major kind of global health problem. We spend billions and millions of dollars on enhancing cognitive function in the elderly so that they can maintain their standard of living. And so this is clearly a very important area of research and has been the major focus. So I'm going to talk about two different studies. Larson looked at 1,740 adults over 65 on the frequency of their physical exercise. And these are more indirect studies. These are correlational studies, but still very valuable. So you go and assess 1,740 people, and basically they're elderly, and you ask them, how many times a week did you exercise over your lifetime, on average, you know? And then you correlate what their answer was to their level of developing dementia. Do you have dementia? Once you do the study in 5 years, how many of them develop dementia? And what is the relationship between the development of dementia and how often they exercised? Oh, sorry, before I do that, the answer was they found that if you said you exercised 3 times a week or more during your lifetime, this includes during your age, you had a 30% less likely chance to develop dementia. Only if you said you exercised 3 times a week or more. Okay? That, you should all be writing that down because that's very important. Because that means that today, what you're doing can affect your probability of developing dementia. Now, Abbott assessed even more men, 2,257 men, aged 71 to 93, on speed and distance of daily walking. This study was done in Hawaii, where they could walk year-round, and so they could assess it all year round. And here again was the same kind of finding as Larson. That is, here they found the larger distance and the faster the men said they walked, the lower probability they had of developing dementia. And again, these men were up to 93 years old. And the older ones that were not developing dementia had a much higher, significantly higher probability of walking both longer and faster than the ones that did develop dementia. But that's a little bit less satisfying because there could be multiple reasons why there was these relationships found. Why? Because we can't be absolutely certain that the people that said they exercise 3 times a week did exercise 3 times a week. There's no way to confirm that. Maybe the ones that don't develop dementia have a bigger imagination and think that they exercise more, and that thought actually influences their cognitive function. That could be the case. This was not a controlled study. It was a correlational study. So what you want to do is go in and actually do a specific study, make patients your experimental subjects, and control as much as you can to ask what exercise is doing for cognitive function. And that is what Colcom did in 2006. Here he had, again, elderly subjects between 60 and 79 years old. Half participated in a 6-month aerobic exercise program. So these were older people. These people were not doing, high intensity, 80% of maximum heart rate on the treadmill. They were walking around a track. They were getting their heart rate up a little bit. They were breaking a little bit of a sweat. This was not high impact aerobics, but it was still getting their heart rate up. And half did toning and stretching. What they did is they used the best tool that we have to study live human subjects, that is the structural magnetic, structural MRI, structural magnetic resonance imaging, and they looked at the quantitation of different brain areas in the 6-month exercisers and the toning and stretching group. And what they found is aerobic exercise participation resulted in significant increases in brain volume in the frontal lobes compared to the toning and stretching groups. Okay? Now, I'll add to this a paper that was just published about 2 months ago, in which they did a year-long, it was the same group, did a year-long aerobic exercise intervention with a same age group kind of people, and they expanded this. They not only looked at the frontal lobe, but they were particularly interested in the hippocampus. Why? For all the reasons I just told you, because we know that there's increased neurogenesis in the hippocampus, and exercise is supposed to increase that. They found significant increases in hippocampal volume, even in these elderly patients, after a year-long exercise program. This was pretty strenuous. They had to call in, they had to check in, they had to do it 3 times a week. There was a certain level they had to maintain, and there was oversight of all this exercise. I can tell you from doing exercise studies myself that it's not easy to get people to conform to these kinds of things. Has anybody in this class done my exercise study where you do bicycle riding in the gym for an hour? Anybody volunteer for that? Okay, well, if you did, there's a lot of drop-off. So even an hour of bicycle riding, even if I give you $50, some people just don't want to do it. And so imagine a whole year of 3 times a week aerobic exercise. There's drop-off. And so this was this study and the new one where they saw increases in hippocampal volume are both very important, very difficult to do studies. Now, 2 more, no, actually I'm going to jump to, so we won't cover this study. But what I'll say is, this is a very important general health issue. Can we increase cognition? Can we increase, can we change brain structure even in the elderly? The answer is yes. But you might be interested in not elderly, but in young adults, in normal age adults. Can exercise actually enhance, change our brain structure and enhance our cognition? And so here I want to just go into the studies that I've done here at NYU. But before I do that, I should really go over some of the reasons why you might see increased, so I'm going to go this over at the end, sorry. We are going to talk about the exercise studies at NYU. So these are studies that I've become interested in the last couple years because of all the studies that I just talked to you about. Increase in neurogenesis. Exercise alone is causing all of these structural, chemical, and functional changes. Can we take advantage of that in me? Can I get smarter? Can I improve my hippocampal function? Can I remember more for my brain and behavior final cumulative exam if I do exercise? So to ask this question, I actually did a study associated with a class that I continue to teach. It's an elective, upper division elective course in the neuroscience major here at NYU. And the class is called Can Exercise Change Your Brain? And neuroscience undergraduates take this class. So it's taught, this is my first year taught in a small room. The more recent classes are taught in the big NPR multipurpose room in the third floor of the Palladium. We can get 30 to 35 people, maybe 40 if we squeeze in there. But this is a real elective class, and it starts out with an hour of aerobic exercise that I teach the students, followed by an hour-and-a-half lecture on the effects of exercise on the brain. So in that class, we go over many, many more studies associated with all the studies that I've just described for you. But we go over the historical studies of neurogenesis. We go over all of the early studies of enriched environments. And then we go over in much greater detail all the human studies looking at the effects of exercise in the elderly and young adults. But I realized that these students could be my first experiment. All I had to do was test them cognitively at the beginning and at the end of the semester. And they were exercising more. I had them do exercise logs. And they also got to get involved in the analysis themselves. So they, as part of their final paper, not only had to come up with a new experiment to study some aspect of exercise and the brain that we went over in class, but they had to analyze their own data from the class. Of course, all the names were taken away. And they compared their data to a control elective class that did not exercise during the semester. So what we found, so first let me just tell you what the experimental design was. This is Can Exercise Change Your Brain class. Cognitive testing on a variety of different tasks that tap hippocampal function, that tap mood, that tap frontal lobe function. Then a whole semester of an hour of aerobic exercise, increased exercise. This was only once a week, but some of the students didn't have a lot of aerobic exercise in their schedules. Followed by a lecture discussion. At the end of the semester, we would test them again. And then they had to do their final paper analysis. And we compared this pre versus post cognitive testing with another control neuroscience elective course. This was a course on molecular neurobiology of learning and memory. They did not exercise during their class. And we also tested them cognitively at the beginning, at the end of the semester. So what did we find? We found that my class, but not this control class, actually improved on one test of hippocampal function. It was a memory encoding task. That meant that my class can encode new memories better and faster than the control class. This was pretty striking because there were only 17 students in my class. It was only once a week. Yet we found a significant difference. So my question for you in the last 2 minutes, that was the first stage of the studies that I'm doing in NYU undergraduates to ask, can exercise change your cognitive performance? Now I want to study the underlying brain changes involved. What do I do? What would you do? What would be your hypotheses? How would you test that? What kinds of questions would you ask based on what we just talked about? What would you want to know? Yes?
Whether the cognitive testing for the improvement of campus function was due to their excitement and enthusiasm in the class, or like that, if their enthusiasm played a role.
Okay, whether the enthusiasm played a role in any change. But let's just assume for a second that there was a behavioral change associated with exercise. Okay? Now what I want to get at is the brain changes that you might look for in this situation. Yes?
Second question. Also, since also in your lecture you would explain how exercise can actually have function.
Yeah.
Wouldn't they also have to believe that they would do better, so they would have to do better kind of a placebo effect? So even though they perhaps they weren't doing better, they were specifically because they thought they were doing better.
Mm-hmm. That could be one. So yes, those two points are asking whether just the thought that they might do better because they're learning about it could change it. But I really want to focus on the brain changes. So we've just spent the whole semester talking about different ways to understand the brain changes. I've told you that the task that improved is one that is memory encoding. What would you look at in the brain? If I could do anything to these poor 17 neuroscience majors and study their brains, what would it be? Yes? Perfect. Yes, she'd look at the hippocampus and the new dendrites forming there and ask whether there might be structural changes, more dendrites, bigger hippocampus. What else? What else would you look for? What else did we talk about in the beginning of the lecture? Yes? Increase in blood vessels, right. That could be another structural brain change associated with this behavioral change. What else? Yes?
Increase in cortical thickness?
Increase in cortical thickness, yes, absolutely. What else? Yes? Dendritic branching, perfect. Anything else? Any factors? Yes? Increase in neurogenesis, perfect, because increased exercise is supposed to enhance neurogenesis. So you might expect increase in neurogenesis. One more, what factor do we talk about that changes with exercise and enhancement? Yes? BDNF, okay, good. So those are all the kinds of things we want to look for. And this is one of the questions that I asked you in the practice questions for this class. So think about that. Yes, one more question? Yes, it's unclear, but what you could do is measure peripheral BDNF as an indirect measure of central BDNF, and you can do that through the saliva. So that absolutely is a non-invasive possibility, but that's a good question. Okay, great. I'm here. If you have any questions, come and ask me, and we'll go over frontal lobe next time.